Good morning, everyone, and welcome to the webinar on the Japanese Knotweed organized by SOILveR. I'm Marta Popova, and I'll be the moderator for the webinar today. I'm here with my two roles. I'm a project manager at the Operational Research Department at SPAC Company, which is a main actor in the brownfield and landfill redevelopment and the soil management in the Walloon region in Belgium. And we are also a member of SOILveR. So I'm very delighted to have you all here. We have an exciting lineup of presentations and discussions planned for today. So let's get started. First, I would like just to precise some technical instructions for the smooth running of this webinar. Please, during the presentations, keep your micros and video on mute. And if you have some questions, please use the dialog box on the bottom of your screen. Raise your hand, or you can simply add your question into the chat. So this will be really helpful to easily manage the questions after each presentation. I'd like also to inform you that this webinar is recorded. So if some of you have some objections, please let us know now. So I imagine that there are no objections. So let's introduce you the subject and why we are organizing this webinar in SOILveR. And why we tackled this problem. The Japanese knotweed is a resilient invasive weed species which spreads vegetatively via rhizomes. It grows very quickly and releases toxic substances into the soil that inhibit the growth of the other plant species. The dense clumps they form choke out and eliminate the native species. It was originally introduced from Japan as an ornamental plant in the Netherlands in the 19th century, and now it's spread widely in Europe. Although it's not on the list of the invasive alien species as established by the European Union, it was actually excluded from the list because it was considered unlikely that its inclusion would effectively prevent, minimize, or mitigate the harmful effects at a reasonable cost on a European scale due to their very widely geographical spread and due to the difficulties inherent in its management. Therefore, the countries that are represented in SOILveR, in Belgium, Wallonia, France, Sweden, and the Netherlands, we consider that it's really an issue for our countries, and that is why we decided to organize this webinar, during which experts that are active in this area of the Japanese knotweed management will share their experiences and their opinion on the best ways on how to deal with this invasive species. So what we have on the program today, we will continue with a short introduction of what is SOILveR, presented by Yvonne Olsen. And then we'll continue with the presentations that are split into three parts. The first part is the detection methods of the Japanese knotweed. The second part is the treatment methods presented by speakers from France, from Belgium, from Sweden, and an accelerated verification and insurance of the Japanese knotweed that will be presented by Paul Bardos from UK. And we'll finish this part of the presentations with one project about the valorization method of the Japanese Knotweed presented by Emerance Bietlo from Belgium. The second part is dedicated to an open discussion between the participants on what we are missing to tackle the problem, and so to exchange, of course, your opinions and point of view about the gaps between the knowledge and the practice on the different methods of the treatment of the Japanese knotweed. And we'll, of course, conclude this at around 12:30, sorry. As you probably have noticed, we have one change in the initial program. Unfortunately, due to health issues, Professor Chris van Dijk from the Wageningen University from the Netherlands couldn't join our webinar. But instead, we have one very interesting presentation for the evaluation of the heat treatments coming from Sweden, from Halmstad University. So we'll continue with the presentation of the SOILveR platform by Yvonne.
Thank you. Then I will share my screen. Yes, thank you. Presentation here. Yes, I will. My name is Yvonne Olsson, and I work at the Swedish Geotechnical Institute, and we are a member of SOILveR. I will tell you a little bit about SOILveR. That is a network that started in 2018, but I would also like to say a little bit about the history, because SOILveR actually is a continuation of the Snowman network that started already in 2003, and which was a funder of research projects and the knowledge exchange network. It started as an AeroNet and continued as an AeroNet until 2009, and then it continued as a self-financed network. In 2016, the European program Horizon funded project that was going to develop a strategic research agenda on soil and land management. And during that time, the Snowman network had a little break or a low profile period to wait on the results for the results from that strategic research agenda. And then in 2018, we started new and had a look at the new information from this research agenda and formed the SOILveR network. And today we are these organizations that are from several countries that are the core of SOILveR, but we also collaborate a lot with other networks, and we are always open to new members, as I will tell you. So what is SOILveR? Well, we are a self-financed platform, and we work with knowledge development and knowledge exchange on soil and land management. We have originally worked a lot on contaminated sites, but as you see with this seminar today, it's not only related to specifically contaminated sites and chemical contamination, but now also invasive species, as we talk about today. What we see is that when we get together across the borders, that we can join forces and join efforts and also finances to actually solve questions or work on questions that are of common interests for us, for the SOILveR members and our countries. We also try to facilitate collaboration between experts and policymakers and other stakeholders to actually take things a step further when it comes to methodologies and also tools and guidance to reach better management of soils. In SOILveR, the members, we set the agenda, the topics that we want to work together, just as this topic today. And we also have a platform secretariat that supports our activities. We have what we call two pillars. We have the knowledge exchange pillar and the knowledge development pillar. And within the knowledge exchange pillar, we have developed several different tools to our support. And these are, for example, the seminars we have today, but we also have shorter exchanges that we call coffee breaks, with more like one presentation on a topic. And we also have internal member expert exchanges on specific topics. And I will tell you a little bit about a work group that we started recently that is called on bioaccessibility and human exposure. That is within our knowledge exchange pillar. We also have the knowledge development pillar, in which we, for example, can launch calls if and when members have an interest of doing so. So it's voluntary for members. And the projects have partners from several countries, but the members, they fund researchers only from their own country, which actually makes it less administrative than if you have than we have with the regular EU calls. We have also started projects among each others. We discuss every year what topic we are going to work on and decide upon that. And we have, as you can see here, a lot has been centered around polluted soils, but it's also broader when it comes to soil and soil health and land management. This is a long list, but it's just an example of activities that we have done these years since we started in 2018. And you can see in 2019, we had a call and we financed 3 projects on emerging contaminant, microplastics, and on recognizing soil values. So that is one example of what we're doing. The other example is that we, for example, in 2021 had a coffee break meeting on oral bioaccessibility that caught a lot of attention. And from that, we also continued with an expert exchange. And there was a discussion that we should have an expert group or working group on bioaccessibility and human exposure. And that's in 2023. I think there's someone didn't turn off their audio.
Yeah, Mr. Geritian, could you please turn off your micro?
Okay, now it's yes. So we started this working group, which is actually running right now, and also in 2024 we actually supported also a research project, because the Swedish EPA was launching a call related to this topic with human exposure and bioaccessibility, and the Swedish SGI applied for a project there, but also involved participants from this expert group that we have. And also SOILveR has been facilitating or is facilitating and is supporting this project just because it's of interest to the members. So these are examples, and you can also see more on our website. And if you are interested in becoming a SOILveR member, you can also have a look at the membership agreement and secretariat fees on our website, where you can also see the previous webinars and seminars and coffee breaks, because they have also been recorded. So that was a little introduction about SOILveR.
Thank you very much, Yvonne. Are there some questions to SOILveR platform? Or we can continue with the first part of our webinar, which is the detection methods. And we have here one presentation for the environmental DNA detection presented by Aurélie Bonin from the French startup Argali. Ms. Bonin, it's up to you.
Hello, everyone. So can you see my screen? Is it okay?
Yes.
Okay. And can you hear me properly?
Everything works fine.
Okay, so let's go. Hello, everyone. and thank you for giving me this opportunity to talk today. So my name is Aurélie Bonin, and I work for a little company called Argali, which is specialized in biodiversity surveys using environmental DNA. We are not specialists of Japanese knotweed, although we are interested in this species because of the ecological and economical challenges it poses. So I will start with a few words on environmental DNA, because I guess that maybe some of you are not familiar with what it is exactly. So here you are. So what is environmental DNA exactly? You have to know that every living organism leaves DNA traces in its environment. It can be under the forms of living cells, of gametes, of mucus, urine, decaying tissues, and so on. And we can collect those traces of DNA and use them for biodiversity analysis. So environmental DNA can be defined as a complex mixture of DNA fragments, more or less degraded, and that originate from a wide range of organisms. So if you go and collect an environmental samples, it can be water or soil, you can use molecular techniques to extract the DNA from this sample, and you end up with environmental DNA. And now, how can we extract biodiversity information from this complex mixture? There are two main strategies that I will detail in the next slides. The first one is based on DNA sequencing. It's called DNA metabarcoding. So from your complex mixture of environmental DNA, you can choose to focus on only particular species using for example, you can use a generalist marker that will target only the Polygonaceae. I chose this family as an example because it's the family that includes the Japanese knotweed. So using this marker, you will focus only on the Polygonaceae DNA fragments in your mixture. And this marker can be seen a little bit like a seeker head in your mixture. And then here you will focus only on the violet, green, and orange fragments. You amplify and sequence those fragments. And based on the sequence, you can compare them to a sequence database. And it will help you identify the organisms that left this DNA. In practice, you end up with an information that looks like this. You have a table with a species, one line for each species you have detected in the mixture. And for each sample that you have analyzed, you end up with a number of reads for the species in the sample. For example, as you can see, there are more than 63,000 reads of Japanese knotweed in sample 1, much less in sample 2, and not at all in sample 3. The second strategy is called, it's based on qPCR, and it has been popularized a lot with the COVID crisis. In the same way that you can detect the COVID virus in your nose, we can detect a particular species of interest anywhere in the environment using qPCR. So in practice, you start with the same mixture of eDNA, but this time you use a specific marker that will target only the species of interest. Here, the species that left the green fragments. And you amplify this DNA, and you monitor the amplification in real time in your reaction. So if the amplification curves go above the detection threshold, you say that the species is present. And if it remains below the detection threshold, as happened here with the black line, you say that the species is not present in your sample. And then you end up with a very simple table, where you say that there's presence or absence of your species. So this is for the two main strategies. Of course, the two approaches have pros and cons. If you put yourself in the context of Japanese knotweed detection, I can give you some limits and advantages. So both of the methods are non-invasive, which means that we don't damage the species. We don't need any botanical expertise in the field. And also you can go and collect samples at any season, which is different from the traditional botanical surveys that need the species to be apparent to be detected. Among the limits, both of these methods are sensitive to contaminations. It means that if you are not careful, and if you don't decontaminate your sampling gear, or if you are not careful in the lab, you can contaminate your samples and create false positives. But also, metabarcoding has its own particularities. For example, for Argali, it's fully operational for Japanese knotweed. This is one advantage. Among the limits, it gives only a semi-quantitative signal based on the relative abundances of reads in the samples. And it's also more expensive than qPCR. On the other side, qPCR is cheaper, as I said, and it's also quicker than metabarcoding. You can get the results in a few days in theory. It also gives a quantitative signal. You can relate you can have a quantitative assessment of the quantity of eDNA in your samples and relate this to maybe the biomass of the species. The literature also says that the detection rate is higher for qPCR than metabarcoding. Personally, I would like this to be proved for Japanese knotweed. I'm not fully convinced, but we'll see. Among the limits, it's not completely operational right now for Japanese knotweed. We still have some technical issues to fix. And there's a higher possibility of false positives compared with metabarcoding. So this is for very general information on eDNA detection of Japanese knotweed. Now I will show you some early results we got on the Japanese knotweed and eDNA. So the first case study I will present today is based on water. And more exactly, we wanted to answer the question, can we detect Japanese knotweed eDNA from rivers? So why focusing on rivers? It's because Japanese knotweed is known to disseminate very well on surface freshwaters. So when it rains a lot, when there's a flood, the rhizomes get torn apart from the plants, and they can travel down the river and disseminate along the banks. So if maybe if you go and sample at downstream, we can have some information on what happens upstream if there's presence of the species. But also we wanted to know if there's some kind of influence of the distance to the closest knotweed patch, if the weather conditions also have an influence, and maybe also the size of the patch upstream. So for this case study, we've collaborated with Aquabio. It's a society full of experts in Japanese knotweed. And incidentally, Mireille Boyer from Aquabio will talk about the Japanese knotweed today later on. So they helped us to design the experiment and choose the sampling sites. So we came and chose 3 different rivers. So it's in the Rhône-Alpes region in France. These 3 rivers are different in the sense that for one of them, there was a big patch of Japanese knotweed upstream, a patch of 150 square meters that's the surface of the patch. The second river, there was a smaller patch of 50 square meters. And the last river, there was no patch at all. And for these 3 rivers, we selected 3 sites. And these 3 sites were at different distances from the patch along the river. And then for each of these sites, we sampled 3 water samples. So 2 on the banks of the river and 1 in the middle. But for all those rivers, they were they were streams more than rivers, actually. So we did this sampling twice under 2 weather conditions, after at least 2 days of sunshine and after 2 days of rain. And here you can see a picture from the field sampling. So it happens this winter in very freezing conditions. And so this is our gear to sample the water. We used 3 different proxies to measure the detection rate. For the metabarcoding, we used the number of sequence reads. So that's the number they were in the tables that I showed you. As we do also technical replicates, it's possible to count the number of positive PCR replicates. This is important because in case of rare DNA, stochasticity implies that sometimes you can capture the DNA fragments and sometimes not. So by counting the number of times where you capture the fragments, it gives you an idea of the quantity of DNA in the initial sample. And the same thing for the qPCR approach. Here are the results for the metabarcoding. So based on sequencing with the 2 proxies. On the left, the number of reads that we detected, and on the right, the number of positive PCR replicates. On those figures, the big patch is symbolized by the dots, and the triangles refer to the smaller patch. For the patch, for the river where there was no plants at all, it's absent from the figures because there were no detection at all. The colors refer to the weather conditions. So orange for the sunny conditions and blue for the rainy conditions. And as you can see, for the bigger patch, you can see a decrease of the detection for both proxies when you go further away from the patch. For the smaller patch, there is some a background level of detection, although it increases a little bit when you are very far away from the patch. We still have to explain why. So if we test those results statistically, we see that for both proxies, there is a significant negative effect of the distance to the patch on the detection level. There is a significant positive effect on the patch of the patch size on the detection. And there's also a significant positive effect of the sunny conditions on the detection. So actually, it's better to sample when it's sunny after several days of sun rather than after a big heavy, rainy period. As regards the qPCR results, they go into the same direction. So it's the same color codes and symbols. And as you can see, there is a decrease of the detection with the distance. And if you test those effects statistically, There is a significant negative effect of the distance to the patch on the detection rate, and also a significant positive effect of the patch size on the detection. The weather condition is not statistically significant. So to sum up those results, it looks quite promising for both methods, but it was a very small study. So we need to confirm the results with more samples. We also like to try to go and sample during the growing seasons where the plants are more active and they can also release leaves in the environment and also decaying tissues. And of course, we need to evaluate the sensitivity for both approaches using more technical replicates because it's quite difficult to compare them right now. And this is our results for the detection in water samples. Now you are probably interested on knowing what happens when you sample soil and what does it mean actually. So we know we are able to detect a Japanese knotweed in the soil when we go and sample at the foot of a plant. Well, that's not revolutionary. I mean, if we see the plant and we detect it using eDNA, there's no point in using eDNA for this. Actually, what you need to know is that in soil, eDNA can persist longer. It can persist several months and even maybe years in some conditions, very cold conditions. And also it doesn't diffuse in the environment. It means that here on this figure, you have a map of a plot. It's a 100 meter by 100 meter plot. And on this map, there's a dot here. The red dot, it represents a tree. So it's not a Japanese knotweed, it's a tree. And we have sampled every meter on this plot, every 10 meter on the plot. And here is where we detect DNA from this tree. So it means that DNA remains very localized. It doesn't diffuse, and this has very big implications for the sampling design and sampling efforts if you want to detect a species in soil. So to answer those questions, the first step was to plan an experimental study. So it's what we call the mesocourse. We did that in a lab in collaboration with Chrysalid, which is a living lab specialized on industrial lands. And so what we did for this study, we collected rhizomes in the wild. And we put them, so we calibrated those rhizomes. They had the same biomass. And we kept them in controlled conditions in a big piles of soil. And for several days, we kept them for 7 days. And then we went and remained for some of them in the middle. We took out the rhizomes prior to the sampling periods. And then we sampled after 7 days and after 14 days. And the idea was to answer the question, is it possible to detect the samples even if the rhizome stayed only 7 days? And how long does the eDNA signal persist? So for now, I can't give you the results because they are still being analyzed. And there's another part of this experimental study. It's the part B. So here we wanted to answer the question about the sensibility of the system. So here we kept different rhizomes of different biomasses, so big rhizomes, medium rhizomes, and small rhizomes. And we kept them in controlled conditions. and we will use, so this is not done yet, we will use the sampling, the optimal sampling after the optimal number of days to be sure that we will detect the rhizomes. So this is still in progress, as I said. So I will, to conclude with on the use of eDNA to detect Japanese knotweed. So for aquatic DNA, the sampling are promising. For soil, it remains quite challenging, and we need to work on efficient sampling designs for soil. Regarding the qPCR assays, they still needs to be improved, but they are interesting because they are quick and cheap. So maybe we can imagine that qPCR could be used for a rough information, and if there's a detection, we can come in a second time and use metabar coding. And what is interesting right now is that technology is going very fast, and there are new developments in the sequencing field, and in particular, the development of the MinION sequencing, which can give results in a few days. So it could help decrease the turnaround time of metabar coding and make it a very interesting tools for managers. And regarding the perspectives, we can also use eDNA for monitoring the efficiencies of control measures. But for this, we need to explore the quantitative nature of the eDNA signal. And so this is the end of my talk. And I would like to thank all these peoples which at some point participated to this project. So all these people in Argali, and also Léa Mention and Mireille Boyer in Aquabio. And also I would like to thank Cécile Grand, which is here today, who actually invited me to this webinar today. And that's it for now.
Thank you very much, Aurélie. There is a question here for you. Bram van de Pas was wondering if you are aware of project of standardizing qualitative Japanese knotweed analysis in soil with eDNA by the Organization of Dutch Commercial Labs.
No, actually, no.
Okay, so probably you can...
Maybe I can, yeah, we'll look into
get
that.
in touch, yeah. Okay, is there some other question for this? For Aurélie? No. Okay, so now we can move on to the second part of our webinar, which are the presentations of the different experiences on the treatment methods of the Japanese knotweed management. We remain in France with the first presentation that is an overview of different known patent techniques that were tested on different sites in France, given by Mireille Boyer from Aquabio in France. Actually, this will be a recording in French, and with the text of the presentation is in English, and it's subtitled in English. And the speaker, Ms. Mireille Boyer, is here today. So if you have any questions, of course, you can ask her directly. So Ingrid, could we share the recording, please?
Alors bonjour, je m'appelle Mireille Boyer, je suis responsable technique de l'activité d'ingénierie de conseil sur les espèces invasives au sein de l'entreprise Aquabio. J'anime également le groupe de travail sur les espèces végétales invasives au sein de l'UPGE, qui est la Fédération française des entreprises de la filière du génie écologique. Je vais vous présenter un panorama des techniques éprouvées et actuellement utilisées en France pour gérer les renouds asiatiques. Je parlerai uniquement des techniques non brevetées et pour lesquelles de retours d'expérience détaillés et argumentés ont été rendus publics. Alors, avant de se lancer dans sa gestion, il est essentiel de bien connaître la biologie de cette plante et il me semble utile de vous donner un aperçu de ses caractéristiques principales. La plus connue de celles-ci, c'est le gigantisme de ces tiges herbacées qui meurent à l'automne et sont renouvelées chaque printemps, produisant ainsi une biomasse aérienne qui est équivalente ou supérieure à celle produite en culture intensive. Mais celle-ci n'est rien par rapport à la partie souterraine, qui est souvent 2 fois supérieure et est constituée essentiellement de rhizomes. Ce sont des tiges souterraines qui accumulent les réserves de la plante et permettent la repousse très rapide des tiges au printemps. Par ailleurs, des fragments de tiges peuvent repousser, comme on le voit en haut à droite, avec cette plantule ici d'une tige arrachée et transportée par l'eau. Mais la dispersion de cette plante est principalement assurée par les rhizomes, qui sont en très forte densité près de la surface du sol, donc facilement dispersables par l'eau ou les engins. Cette plante a aussi un chevelu très dense de racines fines poussant sur les rhizomes et de grosses racines qui peuvent descendre profondément dans le sol. Les fragments de racines ne peuvent pas reformer de plantules chez les renouées, mais elles sont souvent confondues avec les rhizomes, ce qui peut générer des surcoûts très importants lors des travaux. Cette connaissance du volume précis de sol colonisable par les renouées, elle est essentielle pour bien les gérer. Dans ce schéma, on a des indications précises sur l'étendue spatiale maximale des rhizomes dans le sol, qui a été observée lors d'une quinzaine de chantiers et avec plusieurs dizaines de sites traités à chaque fois. En profondeur, les rhizomes ne sont jamais descendus au-delà de 1 mètre environ dans les sols naturels. Par contre, l'extension latérale, elle peut être plus difficile à gérer. La densité de rhizomes, elle est importante jusqu'à 2 voire 3 mètres au-delà des dernières tiges du massif, et on a observé quelquefois des rhizomes erratiques assez superficiels pouvant s'étendre jusqu'à 6 à 7 mètres de distance des dernières tiges. Alors voici un exemple d'une coupe de sol envahie par les rhizomes. Ceux-ci forment un chevelu hyper dense et compact qui occupe dans ce sol 86% du volume et 63% du poids sec. Alors on voit que quand on a des sols envahis qui sont terrassés ou éradés naturellement, chaque mètre cube de terre peut contenir plus de 80 à plus de 600 fragments de rhizomes. Ce sont des données qu'on a observées sur des chantiers de terrassement. Enfin, un mot sur la reproduction sexuée de cette plante. Elle est gynodioïque, c'est-à-dire que certains clones portent des fleurs qu'on dit mal stériles, car les étamines ne sont pas fonctionnelles, et d'autres des fleurs mal fertiles. Les akènes tombent principalement à proximité de la plante et peuvent être dispersées par l'eau. Les graines ont une durée de vie courte et les semis sont très vulnérables. La plupart ne survivent pas à la première saison végétative. Vous avez une illustration de ces semis de renouée sur une rivière. Ils sont rouges et minuscules, quelques millimètres seulement, et beaucoup moins développés que ceux des autres plantes. En fin de saison végétative, ils avaient tous disparu. Alors, toutes ces informations sont importantes à prendre en compte pour définir et prendre les précautions indispensables pour ne pas disperser involontairement des propagules de renouée lors des chantiers. Elles se traduisent par des mesures très concrètes et efficaces, comme la protection des sols ou le nettoyage des engins et des outils qui sont utilisés. Il existe en France plusieurs techniques de gestion des renouées qui ont été développées de manière empirique et elles ne répondent pas forcément au même objectif ni aux mêmes contraintes d'application. Donc il est souvent difficile de répondre simplement à cette fameuse question « est-ce que ça marche ?» On peut d'abord classer ces techniques en 2 catégories. Celles qui s'appliquent à des massifs en place non remaniés et celles qui s'appliquent à des propagules végétatives issues de la dégradation de la plante par des terrassements. Dans le premier cas, vous aurez compris qu'on essaie de gérer une plante sans toucher à son réseau très dense de rhizomes alimentés par ses racines. Si on veut la détruire, il va falloir empêcher la plante de produire de nouvelles réserves et attendre que cette partie souterraine dépérisse. Dans le deuxième cas, on n'a plus des plantes à traiter, mais des propagules végétatives de celles-ci, qui pour survivre vont devoir refaire des racines et des tiges aériennes. En empêchant ces propagules de pousser, on provoque à terme leur mortalité, et cela beaucoup plus rapidement que dans le premier cas. Alors, on voit ici une plantule issue d'un fragment très petit de rhizome de renouée, et on voit que quand ces fragments végétatifs trouvent des conditions favorables, c'est-à-dire un milieu aéré, un peu de substrat, de l'eau, du soleil et des températures suffisantes, ils vont redonner en quelques jours seulement une plante avec ses racines et ses tiges. Mais si ces conditions ne sont pas présentes, le fragment de rhizome va finir par mourir par dessiccation ou pourrissement, et cela va prendre un temps plus ou moins important. Voilà, cela m'amène à ce schéma qui présente les techniques existantes et utilisées couramment en France, classées selon d'une part leur objectif et leur cible, soit la plante entière dans les cases jaune-orangé, soit les propagules dans les cases plus violettes. Et en fait, on constate qu'il n'y a pas moins d'une douzaine de façons de gérer les problèmes liés à cette plante, selon si on veut éviter l'implantation de propagules sur un nouveau site, réduire leur impact écologique ou paysager, bloquer leur expansion latérale, éviter leur repousse dans des stocks de déblais, éliminer les propagules dans des terres envahies, ou enfin utiliser des techniques très longues pour dévitaliser les plantes sans les déraciner. Vous avez aussi dans ce tableau le nom des entreprises et des personnes qui ont mis au point certaines techniques particulières et à quelle date. Je vais illustrer simplement 4 de ces techniques marquées par une étoile. Ce qu'il faut retenir, c'est que la préparation, le suivi et l'encadrement de ces chantiers pour pratiquement toutes les techniques sont toujours très exigeants. Alors, première technique très simple et manuelle, le déterrage précoce. Comment éviter que les propagules ne s'installent sur un nouveau site? Tout simplement en les enlevant à la main avec des petits outils de déterrage. C'est une technique indispensable sur tous les chantiers qui vont manipuler des terres envahies par les renouées et qui vont forcément disperser la plante. Ces propagules vont pousser dès que les conditions seront favorables. Elles seront donc facilement détectables et éliminables à la main en inspectant les parcours utilisés par les engins et les zones qui ont été terrassées. À noter aussi que cette technique est utilisée en France sur plusieurs petits cours d'eau et un lac pour stopper l'avancée des fronts de colonisation. Et il a été montré qu'elle était très pertinente sur le plan financier et sur le plan de ses faibles impacts sur le milieu. Alors, la technique du concassage-bêchage, c'est une technique qui consiste à concasser les terres envahies pour détruire les plantes installées et dégrader le plus possible le chevelu de rhizome. Puis, deuxièmement, empêcher, grâce à une bâche étanche sur le tas de terre concassée, la repousse des propagules produites par le concassage et provoquer ainsi le pourrissement ou la dessiccation de ceux-ci. De nombreux essais en grandeur réelle ont été réalisés pendant 8 ans et ils ont permis de déterminer précisément les conditions de concassage et la durée de bâchage qui garantit la dévitalisation complète de toutes les propagules. Il existe une description détaillée de la méthode dans les retours d'expérience du Centre de ressources nationales français sur les espèces exotiques envahissantes qu'on trouve sur Internet. Alors, si les terres sont correctement purgées ou concassées, une durée de bâchage de 18 mois aboutit à une dispersion complète des renouées. On voit les fenêtres qui ont été faites dans la bâche et qui ont permis de vérifier la mortalité de renouées avant de retirer complètement celle-ci. Il y a de nombreuses repousses de peupliers et de saules, car ce sont des terres en bord de rivière qui ont été traitées, mais plus aucune renouée du Japon. Alors, cette technique du concassage-bâchage, elle est très bien adaptée aux petits volumes de terre jusqu'à quelques milliers de mètres cubes. Pour des volumes qui atteignent des dizaines de milliers de mètres cubes, là, il faut passer à des techniques plus industrielles, comme le criblage-concassage, qui a été développé par Christophe Moiroux à la CNR, la Compagnie nationale du Rhône. Cette technique, elle consiste à faire un premier criblage à 0,10 mm, qui permet de valoriser le cas échéant les terres fines, avec une très faible probabilité de présence de propagules de renouée. Ensuite, le reste des terres passe plusieurs fois dans un concasseur à percussion, qui est un engin utilisé habituellement par les carriers pour obtenir différentes granulométries de matériaux. Et dans les conditions de réalisation de ces chantiers, qui ont été faits surtout en été et dans la région Rhône-Alpes, en France, aucune repousse n'a été observée. Il reste toutefois des questionnements quant au transfert de cette technique dans d'autres régions climatiques. Cette technique, elle est décrite dans le numéro 27 de 2019 de la revue Sciences, Eau et Territoire. Enfin, pour finir, une autre technique est possible pour gérer des milliers de mètres cubes de déblais envahis par les renouées. Elle a été appelée le compactage en couches minces et elle consiste à étaler les terres en couches fines et à les écraser par des passages répétés avec des engins à chenilles. C'est la photo à gauche de la diapo. Cela dégrade le chevelu de rhizome et compacte le sol, créant des conditions d'anoxie qui vont empêcher la repousse des propagules. Toutefois, cette technique ne marche pas sur le rhizome superficiel qui reste oxygéné. C'est ce que vous voyez sur la photo de droite, où toutes les renouées qui sont apparues viennent uniquement de la couche superficielle de sol à moins de 20 cm de profondeur. Cette technique n'est donc adaptée que pour des matériaux qui seront ensuite recouverts, en se rappelant que le dépérissement des rhizomes demandera peut-être pas moins de 3 ans. C'était tout pour cet aperçu très rapide des techniques de gestion en France et je vous remercie de votre écoute.
Thank you very much, Ms. Boyer.
Bonjour.
Yeah. So you have shared two internet sites here on the dialogue in the chat. The first one is the informative platform with all the experiences that you had, that you have tested with some of the methods in France. And the second one is the review that you have cited in your presentation. Le deuxième lien, ça c'est le lien vers la publication que vous avez cité dans la
Oui, j'ai mis les deux liens vers les publications qui sont en français.
Yeah, so these
On
are
peut
the
trouver sur
two.
Internet.
Yeah, and ils sont en français.
En français.
Oui, yeah. So both publications are in French, but with the existing tools, you can easily translate this into English, for example. So I hope that you followed this presentation. It was quite clear. Are there some questions for Mireille? Okay. So then I have one question for you, which is what is the optimal monitoring period in order to evaluate the effectiveness of the techniques, of the majority of the techniques? I know that each technique demands some different time slots, but what is the optimal time period?
Je crois avoir compris la question. Pardon. Il n'y a pas de période privilégiée en fait. On peut le faire à toute période.
Okay, so
Pardon,
there are
excusez-moi.
no.
Sauf, ça peut être, c'est plus des raisons pratiques en fait. Quand on travaille sur des sols, par exemple, il y a des conditions pour le criblage, notamment sur la méthode de la CNR de Christophe Moiroux. Il y a des conditions d'humidité du sol, surtout pour le premier criblage, qui sont contraignantes. Donc, qui fait qu'il faudra plutôt faire ça en été qu'en hiver.
Mm-hmm.
Voilà.
So for all the techniques that were presented, there is not the right season to apply them. But for the technique of the crushing that was tested that was developed by Christophe Moreau, the best season might be the hot season in the summer or in the spring, because the humidity of the soil should not be so important, so that the technique is effective. Ok. Et concernant la période de monitoring, après avoir mis en place chaque technique, est-ce qu'il y a une période optimale? Vous avez montré ici avec le bachat, c'est 18 mois après. Mais pour les autres techniques, est-ce que c'est une saison de repousse ou 2 saisons de repousse?
La méthode de la CNR, la mortalité est immédiate en fait. Voilà. Il y a les propagules, il n'y a pas de repousse de propagules, c'est immédiat.
Alors que c'était avec le
criblage concassage.
Okay.
Alors que la première, la mortalité n'est pas immédiate et il faut attendre 18 mois pour avoir le résultat.
Yeah, okay. So with regards to the monitoring period after having applied each of the techniques, for the first techniques, as we saw for the tarping, the monitoring should be 18 months after having put this geomembrane or the plastic. While for the second technique, the devitalization of the rhizomes is immediate, so there is no waiting time in order to evaluate if the technique is effective or not.
Et la dernière, si je peux compléter, la dernière technique que j'ai présentée, le compactage en couche mince, les propagules sont pas du tout mortes. Et on peut penser, bon, qu'il faut au moins 3 ans pour que les propagules dépérissent. Mais le délai n'est pas, on n'a pas fait assez d'essais pour savoir exactement le délai de dévitalisation.
Yeah, and with regards to the third technique, which was the compacting on thin layers, so in this case, the propagules don't die immediately, but normally it takes up to 3 years for the complete devitalization, but it should still be tested and developed in order to better evaluate what is the period. Okay, and here we have one question from Tina Herteveld. What is your experience on the crushing? Is it easy to perform? Concernant la première, la technique de Moro.
C'est celle donc si c'est facile à mettre en place, c'est ça?
Oui.
Là, c'est plus une technique industrielle, je dirais, donc qui demande des moyens importants d'organisation, des grosses plateformes pour faire le traitement. Donc
ex situ?
Oui, en général, oui.
Ouais. Okay, so this technique of the crushing requires machinery, the proper to have the proper equipment, and also it's a technique that is applied ex situ. So you need also to have the proper space in order to deploy this technique. So entre 1 à 5, vous estimez combien 5 étant le plus difficile et 1 moins difficile? Vous situerez à combien?
En fait, je pense que tous ces chantiers sont difficiles à gérer à cause du risque de dispersion de la plante, c'est-à-dire d'avoir l'effet inverse à celui recherché. La technique elle-même, elle est simple. C'est du criblage ou du concassage. Donc ça, en fait, c'est très, très simple. Ce qui est très compliqué à gérer, c'est le risque de dispersion. Parce qu'on travaille avec des engins qui vont manipuler des dizaines, des centaines ou des milliers de mètres cubes de terre. Et le très gros risque sur ces chantiers, c'est de disperser la plante. Et ça, c'est très complexe à gérer. Et plus le chantier va être gros, plus ça va être complexe.
Okay, so it's as Ms. Boyer precise, it's a complex technique to deploy, but what is the most important here is the risk of further contamination with the Japanese knotweed, and the biggest the site is, the biggest the risk is of further contamination due to the fact that you use this equipment and this machine on site, which can lead to a further dispersal of the propagules, right? The propagules of the plant. Any other burning questions? Or we can thank you very much, Ms. Boyer, for this enlightening presentation.
Merci aussi.
And we will continue with the next technique that will be presented by Bart Vermeeren from Hammers Technology Benelux. So Bart, the floor is yours.
Yeah. Good morning, everyone, and greetings from Belgium. Can you see the presentation? It's okay like that?
Yeah.
Yeah. So okay. Thank you very much, SOILveR, for inviting me as a speaker on this webinar, and for giving me the opportunity to share the outcome of our experiments on the eradication of Japanese knotweed by thermal treatment that we executed in partnership with the University of Leuven. So just give you a short introduction of our company and the functioning of our technology. Hammers Technology is a Belgian company based in Brussels, And we develop already for more than 25 years soil remediation techniques based on thermal desorption, which I will explain to you in a moment. We have an extended R&D department, and we produce our burners called smart burners in-house. So we currently hold more than 50 national and international patents, and our technology is licensed and used worldwide, as you can see on this map. But most important, what I want to show you is how does thermal desorption work? So there are basically 3 important steps. In the first step, we start heating up the soil by conductivity. So 2 steel tubes are inserted into the soil, and hot air generated by the smart burner circulates in the first tube in order to transfer the heat to the soil. So first the groundwater is evaporated at 100 degrees Celsius, and then the temperature continues to rise until the contaminant evaporates. So we bring the soil in negative pressure and extract the vaporized contaminants through the second perforated steel tube. In the third step, the vaporized contaminants are treated as fuel in our burners or in a vapor treatment unit. So that's basically how the technology works. And here you can see an example of an in situ treatment in Gela in Sicily. And you can see the specific pattern in which the tubes are placed to make sure that we reach the desired temperature in the complex treatment area, in the complete treatment area. So the tubes are placed in a triangle pattern, and in the center of the triangle is what we call the coldest point. So when we reach the target temperature for, let's say, 220 degrees for hydrocarbons or 350 degrees for mercury or PFAS at these coldest points, we are sure and we know that the whole area has reached the desired temperature. So what are the advantages of working on with thermal desorption? Well, there are several. First of all, and I think it's also an important one in the treatment of Japanese knotweed, is that we can do in situ and ex situ treatment on location. So we don't need to excavate the soil, and when it's already excavated, we don't need to transport the contaminated soil. We can treat it on site, and so everything stays in place, and we just extract the pollution. We can apply our technology in or near infrastructure, so like houses, roads, but also close to valuable trees or other plants without stability risks. So our technology is scalable, as every tube and burner is a treatment unit. So we have already an installation with 3 units, but we can scale up till 350 units. And we only have to bring our tubes and burners to the location, insert them into the ground and start the treatment, which has a limited inconvenience. So there's no noise, no smell, and no traffic during treatment. And as we have a short treatment, we are not talking about years or months, we're just talking about a few weeks, we can dismantle the installation after cooling down. And as we are doing this already for many years in soil remediation, we can, in this particular area, guarantee a clean soil and fixed competitive prices. So let's talk about Japanese knotweed. Why did we start with testing on the eradication of Japanese knotweed? Because, as I just explained, our technology was not invented for the treatment of the plant. The Japanese knotweed just crossed our path by accident when it grew from our neighbor's garden to the parking lot of our offices in Brussels. And people told us that the Japanese knotweed could be treated by destroying the cell wall of the rhizomes. And we thought this could match with the functioning of our technology. So our first concern was, can we ever have an affordable solution? Because, as you know, soil remediation is not always a cheap activity. And we believe this is possible because on the one hand, we learned that we are able to work at low temperature. So we are not vaporizing groundwater, which is normally a big part of our energy cost and also does cost a lot of money. And there are no contaminated vapors, so there is no vapor treatment unit necessary. And these two important elements are missing in our costing on the treatment of Japanese knotweed. So we decided to do 3 experiments, but as told before, we know nothing about the plant itself. We decided to look for help, and we find this by the summer of 2021, when there was a major flooding in Belgium, and certainly in the southern part of Belgium, in the Walloon region. And there was a widespread of Japanese knotweed that was situated on the riverbanks. So by the articles in the newspapers, we learned to know Professor Van Meerbeek at the University of Leuven, who was giving advice on controlling the spread of the Japanese knotweed after the flood in Chaudfontaine. And we contacted him and asked him to supervise and validate our tests. We did exactly 3 tests. Our first test was an experiment, but was on lab scale. Was conducted by 4 bachelor students, bioengineer. And the idea was to fine-tune the methodology and identify the optimal treatment temperature and period and time. So the rhizomes were excavated and exposed to different temperature over different periods in lab conditions. So we sorted the rhizomes in 3 sizes, like thin, medium, thick, less than 1 centimeter, between 1 and 1.5, and more than 1.5 centimeter. And we exposed them to 50 degrees Celsius and 80 degrees Celsius for 1, 2, or 3 days. All the roots, control and treated roots, were subsequently planted in the greenhouse, and we were giving them the optimal conditions to regrowth. And as we can see that in table 4 below, that at 80 degrees Celsius, that already after 1 day we had no regrowth for the treated rhizomes. And for 50 degrees Celsius, 1 day was not sufficient. We saw resprout almost for every size. 2 days, we had some small regrowth for the thinnest one, but after 3 days, there was no regrowth at 50 degrees Celsius. So we learned that or a short period, 1 day at 80 degrees, or a little bit longer at 50 degrees should be sufficient to treat the Japanese knotweed. The second experiment was not longer carried out in a lab, but we carried it out in a 3 cubic meter container and aimed to treat the contaminated soil with our technology with 1 single tube by heating up till 80 degrees Celsius. So the rhizomes were excavated in Antwerp, as you can see on the left. We took samples from different layers, from the layer 030, 3060, and 6090 centimeters. We sorted out in different sizes, from thin, medium till thick. And we had control samples that we sent to the greenhouse to see that it was a viable plant, was well alive, but you can see it also on the pictures. And then the rest of the rhizomes we treated in the container. And after 7 days, we reached the temperature. We kept it for 24 hours at 80 degrees Celsius. And after cooling down, we brought the treated rhizomes to the greenhouse, but we had no regrowth for all the treated items. And the third experiment was an in situ experiment that took place in the Netherlands, more precisely in Heerlen. The target soil temperature was set at 80 degrees for 24 hours and 3 days at 50 degrees Celsius. And we carried out because, as mentioned before, but I learned in the previous presentation, that the rhizomes, they resprout and the roots, they don't. At that time, we didn't have that knowledge. So we carried out a probing to see how deep we can find roots or rhizomes. And we dug also trial trenches to see how far the network will go for the rhizomes. And as you see on the picture on the right below, we see that it has a robust and intensive network below ground. So with our technology, we only target the underground parts of the plant, as the plant is very resistant to treatment actions targeting the above ground part. And in Heerlen, we treated an area of 26 square meter, and we went till 4 meters deep to be sure that we have the complete zone treated for the area. Before the installation of the burners, we removed the above ground biomass of the Japanese knotweed, and we decided to use 8 smart burners for 16 tubes. As we have not a very deep treatment and we have not to go very high in temperature, we reused the warm air in 2 tubes. And after 13 days, we could have shut down the installation. So we reached in every zone or the 80 degrees or the 50 degrees for 3 days. And after cooling down, we took out some treated rhizomes in the layer from 0 to 30 centimeter. And we tried to regrow them in the greenhouse, but we had no regrowth of no resprouting over there. But the most important test is in nature. So at the left side, you can see we chose a rather small isolated Japanese knotweed plant. It was in June 22 that we did the test. And after 1 year, in 2023, we did the monitoring every month. We went to the location to see if we could see regrowth. We made a fence around the treatment area to avoid maintenance by the Green Service Department. And exactly a year after treatment, we saw that there was a lot of regrowth of other plants in the area, but we could not detect any knotweed anymore. So for us, after the 3 experiments, our first conclusions were that as we are specialized in controlled heating, we stick very close to our core business. So we can guarantee that we reach the desired temperature and duration in the complete zone. And we believe that our treatment is environmentally friendly. It means that we heat up by conductivity and don't change the structure of the soil. We stay low in temperature. We have a short duration, so we just stay for 1 day or 3 days at 50 degrees or 80 degrees. And as we add nothing to the soil, there are no residues left behind. As Japanese grows in the most challenging and sometimes impossible places, we need to be very flexible, I think, in the future. We guess it will be most of the time always a tailor-made solution. So depending on the size, we can scale up from 3 burners to 300 burners. We should probably place our tubes vertical, horizontal, diagonal. And we are aware that we should also work in very different types of places. So it will be industrial sites, open spaces, but even on slopes, on riverbanks, or close to infrastructure. So very important from our point of view for treatment of the knotweed is that we go on site, so there is no risk of spreading. Everything stays in place. And we see that the Japanese knotweed often grows in urban areas. So we have a very limited impact because during treatment, there is no noise or odor, or there is no traffic. As we are doing this already for many years, and in soil remediation projects, we have to go to high temperatures. 50 degrees and 80 degrees is for us like starting up. But this is already the treatment phase in attacking the Japanese knotweed. So we are comfortable and we can say that we have a predictable result. And as we can see how quickly nature has recovered above ground, we like to believe that we have little impact. So we aim at in the future to have the technology adapted to treat Japanese knotweed, and that we go for a permanent removal in a short processing time. It means not for weeks or months or years, but in a few days. But still, we have a lot of research questions. Maybe that's also something for the discussion afterwards. As in, for example, in Heerlen, we dig for the rhizomes and the roots, and we went for 2 or 3 meters deep. So what is the best treatment depth to go for? Is there a risk if we treat for example, 1 meter, that there are still rhizomes sprouting once they are separated for the mother plant? What is the optimizing treatment temperature and duration? And we certainly need to work on that, because we see that Japanese knotweed is often close to utility lines like electricity, gas lines, water lines, telecom. And we find it also very difficult to determine the surface of the plant. So are there methods to find out the surface without probing and digging and see where we find the last rhizome? Nevertheless, we saw a quick recovery of nature in the treated area in Heerlen. We want to do some more SOILveR research, what's happening underground after our treatment. And what we see, and especially, or at least in Belgium, we see a lack of legal framework. So as Japanese knotweed is not a contamination or a waste stream, how can we get the validation of the treated soil? So that's still a big question mark from our side. And of course, we want to develop our technology more specific, adapted to the application for treating Japanese knotweed, and maybe combining this also with other techniques. So thank you. This was my part for the presentation. Maybe there are questions.
There is one first question. Thank you very much, Bart, for this very interesting method that you've presented. So the question is if you have an experience in polluted areas, PHAs, heavy metals, benzene, and does the equipment need to be adapted to the pollutant to limit, for example, the risk of volatile pollutant release? I imagine that here we are speaking about the mixed pollution, Japanese knotweed, and on polluted areas. Is it right?
Yeah, I guess that that is a question. If indeed, if we're working in contaminated areas, then maybe probably there is a bigger problem than only the Japanese knotweed. We see that Japanese knotweed has no problems to grow in contaminated areas. So it is very important indeed that if we are aware that we have to do treatment of Japanese knotweed uncontaminated soil, that we have the appropriate conditions and that we also collect the vapors. On the other hand, as we stay below the 100 degrees Celsius, as we're just aiming for 50 or 80 degrees, a lot of contaminants will not be vaporized by at that temperature.
Okay. Thank you. I have another question. What is the impact on other microorganisms and or soil nutrients by the applied temperatures used in this technique? And how far along is this soil analysis pilot?
This is, as I mentioned in the last part of the presentation, this is something we're working on right now. We saw good regrowth above ground, so there was a lot of local herbs growing again, but we still want to examine what's happening below the surface and how the microbiology is behaving. So close to the tubes where the temperature will be a little bit higher, we have probably some kind of pyrolysis process. So we are creating carbon. But we have to see what's the impact, but that's a part of the next experiments that we will do.
Okay. Thank you very much.
You're welcome.
We have another question. What are the treatment costs and how much energy is consumed?
We are not that far. As I told before, we have just did 3 experiments. We are now doing next experiments to optimize the technology. Do we have to go like 4 meters deep? If I understood well in the previous presentation, the roots that are very deep, they do not sprout again. It's only the rhizomes. The rhizomes are more superficial. So there's a big difference in just treating 1 meter or 4 meters. Will there be a different treatment in summer or in winter? From our point of view, normally not on energy consumption because temperature underground is stable. But we don't know because then the energy is into the rhizomes and into the roots. Does it has an impact? So after optimizing the technology, we will be able to give an upfront price for the treatment.
Okay, great. So I suggest to close the questions and to note your question and to leave it for the last part, which will be an open discussion between the participants. So that will keep in the timing. So I suggest to make now a little break, a short break until 11:30 to grab a coffee or some refreshment, and we'll continue at 11:30. Is it okay for you? Yes.
Sorry, Marta, I guess
Now
my
we'll continue
hour was a bit wrong.
the second part of our webinar. We are still in the treatment methods, and this is another project on the thermal treatment methods presented by Tina de Hertelveld from the Halmstad University, in Sweden. So, Tina, the floor is yours.
Thank you, Marta, for the
We
introduction.
can't hear you very well.
Okay, let's see. Do you hear me better?
Probably it's my phone. Yeah.
Is the
It's
sound
yeah,
okay?
yeah.
Good.
That's good.
I'll just find my... So, this also okay?
Yeah.
Okay, fine. So thank you, Marta and Yvonne, for the invitation. And I would also say thank you for Bart Vermeeren for the previous talk, because it was, there will be some repetition, but I would just go through what we have been working on here in Sweden, because there is quite an interest of different heat treatments used to control Japanese knotweed. And it's not just in the lab phase, as you just heard before the break, it's also companies performing heat treatments for different clients. So my part has been to collect material from these different companies and see if it has worked, basically. So we have checked the distribution of Japanese knotweed in Sweden to see if it's interesting in our northern part of Europe, and yes, it is. And then we have, just like Bart said, we have done the same lab treatments with heat. And then what's most interesting, I believe, is the scaling up of the heat treatments and how we should evaluate if the treatments are working. So we're a bit further north, and Sweden is a very narrow and long country, so it's basically a problem for us in the south. or central Sweden, and then we have it along the Baltic coast, but not yet reported from the mountain areas in the northwest. And we would like to keep it that way. Why it's interesting is, just like you have already heard in all the previous presentations, that we have these contaminated soil masses, which then are a huge cost in many budgets. So, for example, this is pictures from construction of a bike path. And we can see that we get quite a large volume of soil that we need to then contain. So here it's just basically put onto a geoweave, which is then sealed, and then we just plant something on top of it. It's of course not the sustainable use of topsoil, which is something we want to keep, and it's also not a sustainable use of lorries and geoweave. So it's a very costly business. That's sort of how we came into this area. What we've been doing for the past few years is to specifically look at the heat treatments. We also started with lab tests back in 2021. So I believe we have been working in parallel pretty much with the Belgian group. And then there are then in situ tests. And I will present from some different companies then who are involved in this. As you have heard, there are thin and thick rhizomes. And the top photograph is a thick rhizome, so they can get quite sturdy, which is interesting when you talk about heat, because the heat needs to penetrate the biomass long enough for all the cells to die. But of course, we also have thinner rhizomes. So the bottom left picture will then show these thinner structures. And shoots can also be interesting, because there are reports that if you chop shoots off and they are put on damp soil, they can also root. But this is a minor problem compared to the rhizome spreading. The evaluation we have done has been a dual approach of cultivation in the greenhouse and then fluorescence microscopy. And fluorescence microscopy is to check if cells are dead or alive, and it's a technique that's been used for many different organisms for tens of years. So it's been used in mice and all kinds of organisms, where you want to pinpoint if a treatment has knocked out the cell. And as you have heard here, 80 degrees is sort of a target temperature. And the reason is that at 80 degrees, proteins will coagulate, and proteins that lose their shape also lose their function. So then the cell walls will also lose their function, and we will have dead tissue, which is what we wish to obtain with heat treatments. Just a short look of how it looks with the fluorescein diacetate staining. That is one of the dyes we have used. To the left, there is a rhizome cross-section where we show a live control, and we see flourishing green cells, which is then live biomass. To the right, we have had a lab treatment, 24 hours, 80 degrees, and we still see fluorescent cells. So this means that 80 degrees may well be a good temperature, but the timing was, our conclusion was that the timing was too short. So we increased it for 8 days to just, conclude if heat at 80 degrees was effective, and we could see, well, yes, it is. If we leave it in 80 degrees for 8 days, yes, the rhizome will die and it will not flourish. There is quite an interest in microwave compared to heat with other methods. And if you put a rhizome in the microwave for 20 minutes at full effect, yes, it is also dead. But you can see there is a faint green shine. So we had some developments. We needed to look into the technique to see what is live and what is actually dead. To go out in the field, we chose to work with or to look into one company, Heatweed, which is well-known weed removal technique. We chose it because it has been used a lot. There were many sites to collect material from, and it had also been used by different municipalities against Japanese knotweed. So you can see that there are little holes in the soil, and this is where you put down a probe and where then steam is put into the soil to kill off Japanese knotweed. So it's hot water, the temperature is over 79 degrees. When we collected rhizomes and grew them in the greenhouse, we checked the proportion of shoots that emerged. And to the left, the three bars to the left are all controls, and they all grew. But then we have samples from 4 different municipalities, and you can see that the effect of heatweed was actually quite varying. If you look at the second one, called Falsterbo, there is no emergent shoots. But if we look at the rightmost sites, Motala, we have everything from maybe 10% emerging shoots up to 75% emerging shoots. So the conclusion of the heatweed was that it gave a very variable effect. It could kill rhizomes, but you can also end up with rhizomes still being there. And of course, the site where you have had rhizomes surviving, it will regrow. The second technique that we were involved in was in situ treatment, also with steam, but where the treatment went deeper down. You have already heard about this issue of how deep down should you treat to kill off all the Japanese knotweed. Inverterm typically goes to 1.5 to 2 meter depth, and the goal temperature is 85 degrees. Here we checked from 3 sites, and we checked this is results from the greenhouse. So what we could see was that from these 3 sites which had a bit varying temperature and a bit varying number of days of treatment, but a minimum of 3 days treatment and a minimum of 75 degrees, we found no regrowth, while the controls grew like they should. So here we can see that compared to the more superficial steam treatment with heatweed, we can see that the Inverterm, we see lower regrowth or no regrowth. We also did on these samples in microscopy, and the gray bars are the interesting part. This is micrographs that are all black. So these are photos of the rhizome that look like the left picture in the top part of this slide, so all black. So 83 micrographs without live cells means that 83% of all the slices we took showed no life. Some parts had small dots, and that's the pink parts of these bars, and that's like the middle photograph. So it's almost all black, but we occasionally see a flourishing cell. We are not clear over how much this actually means in terms of functionality and survival of Japanese knotweed, but to be very transparent, we just diversed between, we split the micrographs that were all black from these that had tiny 1 or 2 cells. But interestingly enough, in the heat treatment micrographs, we had no ones that looked like the right photograph, so no live rhizomes. This was for 2 sites, and then the third site was a bit further on in our cooperation with a longer treatment time. And here we could see that the number of micrographs without live cells had actually increased 1 step more. So it was possible to still adjust this method and get a higher degree of dead tissue. So what we concluded was that if we have really a high temperature and a very long treatment, we also get very high death of these rhizomes. Of course, we are very interested in reducing the number of days that are needed and also the temperature that is needed to make it more feasible to perform these treatments. One company that I haven't, well, we are planning to cooperate, but I haven't been part of so far, but I thought it was interesting to mention, is SOILsteam International, because they also work in situ, but they use excavated material. So this is interesting for sites where you excavate the Japanese knotweed anyway. And there is a picture of their setup and also a closer picture of what is actually treated. And it's like plastic crates with contaminated soil that then is steam treated with short bursts. So here the treatment time is not days, it's minutes. And the temperature is also around 80 degrees. And from that publication, if we take a look at survival, the left-hand graph is of Rhinotria bohemica, so not Rhinotria japonica, Japanese knotweed, but the hybrid. So close enough to be interesting, I thought. They looked at rhizomes that were either 5 centimeter long, so the red line, or 10 centimeter long, and that's the green curve. And if you look at the x-axis, we can see that at 80 degrees temperature, we have very, very low proportion of sprouted rhizomes. So they also took rhizomes into the greenhouse, grew them, and we can see that there is a very low survival. So their conclusion is that if you expose this excavated soil with rhizomes in it to 80 degrees treatment with steam for 90 seconds, then you will have very, very low survival. And then finally, and this is, Bart will recognize this because I will go through this very quickly since you had a great presentation about this technique just before the break. But we cooperated with a different company that's also specialized in thermal conductive heating, and we tested a Japanese knotweed area in Sweden that was 150 square meters. We also had similar goal temperatures and similar depth of treatment as in the previous presentation. So what we did was to put, or they did, to put up this contraption down to 4 meters. And then the goal was to have the goal temperature reached down to 3 meters. This is just a photo of the site. As you can see, it's a slope, and then there's a bike path, and then on the lower end of this slope is a river. So it was quite a complicated area. It also has some interesting geological setup with lenses of different soil types. So quite some challenges in treating this. That's why we chose to cooperate with Krüger, who then came and set this up in place. And we had a long period of treatment. So we started in March and didn't end until in August. This was a trial, so they hadn't done this before, and we hadn't done it before. So you can see to the right, the different temperature measurements of different depths. And one interesting thing is that at the lowest depth, so 2, 3, and 4 meters, that's the upper curves, they reached the goal temperature and it was stable. Once the temperature was reached, it was stable. But we had more problems to heat actually the surface. So the 0 meter is the surface, and we had last year when this was performed last summer, we had heavy rain in this area of Sweden. So that's why we had to insulate with the yellow mats that we put on top. Okay, so what did we see? Well, we could see that it took some time to reach the heating. But once the soil was hot, it was really stable. One other conclusion that we could draw in the field was that it's important to heat the whole area, because in May, so when the temperature was almost 80 degrees, we still had this little patch of Japanese knotweed growing through the insulation mats. So this means that although the temperature probes picked up that it was hot, it was still heterogeneous heating in this slope. So therefore, we let it just heat for a longer period to really obtain hot soil throughout this Japanese knotweed stand. Then we took very careful sampling because, like several other presenters this morning have said, it's important to know at what depth we find the rhizomes. And the pictures are a bit blurred because it was very steamy to stand down in these pits. But in the middle photo, you can see my little trowel. And on the right one, there is a rhizome. So, and that was what we could see, that was our deepest depth, and it was 1.35 meters. So not deeper than that. Also, the rhizomes were very dark, very black, very slimy. So you could see almost from the beginning that probably this would not sprout in the greenhouse. We anyway grew them, and what we could see was that at the end, so the rightmost bar at the 26th of September, there we had no shoots that emerged. In the beginning of the treatment, we did have. So in April, after a month of, heating and problems, and there was one that sprouted, but the other months we found no sprouting rhizomes. So to conclude this short presentation, we see that temperatures of 80 to 90 degrees Celsius seem to be efficient in killing off rhizomes of different diameter of Japanese knotweed. We see that in situ treatment requires long heating period so that all the soil is properly heated to the goal temperature. And we also see that there is an alternative route of treatment that's on excavated material, where they can then be treated with shorter heating periods. So thank you, Marta. That was all for me.
Thank you very much, Tina. This was very interesting presentation also because we have another method for verification. So we have one question here from the audience. And the question is, what energy kilowatt hour per square meter was required to get effectively rhizome destruction?
Yeah, I have some numbers, but I unfortunately do not have them in my head. What I can say is that the different companies use different fuels. So anything from just electricity, obviously, to diesel, to HVO, to even wood chipping is tested. So there is quite a lot of interest in pinpointing this, the cost for the energy, and also to get down. So we have as short time of heating as possible to get the costs and the energy use down.
Okay, I'll take the two next questions because you need to leave at noon.
Yes,
right?
really sorry. Yes.
Yeah. So the next question is there a difference in the results with other soil types, such as sand, clay, and gravel? And can Japanese knotweed survive longer in other soil types?
Excellent question. Yes, definitely there is difference. And the heatweed results where there was eradication at one site, we believe that this is because it was sandy soils where heat will disperse more easily. While the last experiment in cooperation with Krüger, we had these lenses of clay, really heavy clay, which were cold and more difficult to heat. But where there were still rhizomes in this clay, which I thought was interesting.
Okay. The next is a question or more a comment on the FDA and the TCC. Do you consider that these are reliable tests in order to verify the effectiveness of the method? For example, the TCC. Yeah, the TCC reacts with lignin as well, and the FDA.
Yeah, absolutely. So it's about. Am I right? Or no? Oh, just so I get the question right.
Yeah.
Is it about the dyes that we used? No?
I'm not sure, I don't know, Paul.
If it is about the dyes, it's right that they react with the lignin as well.
So for FDA and TTC, these tests were patented some years ago in the UK for knotweed treatment, but they're unreliable. We spent a long time trying to get TTC to work, but it reacts with lignin. We tried to do extracts to see if we could get a colorimetric response. We couldn't do it with at best 90% reliability. For the FDA, our evidence is that will fluoresce with both viable and killed rhizome.
So the TTC I'm not familiar with. We have propydium iodide as the counterpart to the FDA. So
TTC is
I believe we haven't used.
TTC is a different viable stain. That's a red-colored stain.
Yeah, for the red-colored, we used propydium iodide, and there you're completely right that it dyes the lignin as well. So, and I believe that because it should go for the nucleus, so the DNA, RNA, and when we use it for heat-treated material, our hypothesis is that the heat treats, it kills off the nucleus so rapidly so that we will not pick it up with the propydium iodide.
And the fluorescein diacetate, we found that reacts, that fluoresces with cells that we know have been killed, with rhizome that we know has been killed.
Yeah, what we found is that we need to control for the autofluorescence of the Reinutria. So even if you haven't used any dye, there will be some fluorescence. Yeah, so we found that we need to subtract that.
But in terms of the overall pathway you're doing about time temperature regimes, that's definitely the way to go to optimize the heat treatment.
Sounds interesting. Yeah, it was very interesting to listen to Bart's presentation just before. So thanks.
Okay. Thank you so much. There are no more questions for you, Tina. So thank you very much and have a nice day ahead.
Thank you, you too.
Okay. So we will close this second part of the treatment methods with the assessment, with the presentation made by Professor Paul Bardos for the accelerated verification and the insurance of the Japanese knotweed management techniques. So Paul, the floor is yours.
Thank you. I'm going to share my screen. And has that gone to full screen?
Not yet.
Is it on full screen now?
Still not. If you can double click.
Try again.
Yeah.
Is that better?
Yes, that's okay.
Okay. Sorry about that. So this is the material. Can you hear me okay?
Yeah.
This is the plant in question, Japanese knotweed. And before I start, I'd just like to say a bit about PDR3. That's actually made from our initials. So my background is as a soil scientist in land remediation. David and Richard have worked in Japanese knotweed treatment for more than 20 years. They set their company up in 2004. They have written a large part of the UK guidance on Japanese knotweed treatment, which, as you know, is a big issue in the UK. And they have a very strong interest in more sustainable technologies for knotweed treatment than area spraying. For instance, they distribute the stem injection technology across Europe. My colleague David is on the call, and I invite him to jump in at any time that he wants to. So today, I'm going to be quite quick, actually. We'll go through the benefits of viability testing, the testing process that we suggest and the materials needed, the procedure, the results and the reports, the costs, and the warranties that we can supply to go along with this testing. So we've spent about 4 years getting to this point. We have a UK patent and a European patent being finalized at the moment for this viability testing system. We've tried many different types of tests, including the staining tests that Tina was using. A big problem for Japanese knotweed, particularly for germination and greenhouse tests, is that the rhizomes can be shocked into dormancy. So I'll ask David to speak about that a little bit at the end of my presentation. But the long and the short of it is that they may stay in stasis for several years in the ground and then regerminate at some random point in the future. So our goal was to find a viability test that was faster than germination tests, which can take weeks to months, depending on the approach, that was reliable, and that could detect whether a rhizome was dormant and still viable. We have tried this test in 4 laboratories now in the UK, the Netherlands, and Belgium, and it's worked in all 4. It's quick and it's accurate. It's a very simple approach. Well, the analytical part of it is a relatively simple intervention, and the results are available within 48 hours. We've also developed a statistical package to support this testing. While we can determine whether individual rhizomes are viable or not viable, we also think it's important to be able to determine if an infestation has been treated or not. And obviously the costs of sampling every single rhizome that you find are prohibitive. So this statistical map package works on defining what might be an acceptable outcome level. For example, that a very small fraction of 1% of rhizomes are viable, and an unacceptable outcome level. For example, more than 1% of rhizomes are viable. It's statistically impossible to show a 0% viability. So this is why we choose a very small percentage. And the statistical maps, we've just put in notional bars. The red bar going up is the viability threshold, and it's just notional. It's a percentage value, in fact, or a ratio value. The big map, you can see like a blue dot there, and the blue dot is the average viability test result. And then we also can show confidence limit. So there's a dashed confidence limit for 95%, and the blue bar, or the blue perimeter, is 99.5% certainty. So this is enough really to be able to base a decision. So you can, in this large map, where that whole 99.5% blob is underneath the green line. The green line is the acceptable outcome. Then you can say that the materials there have been treated. And for an infestation, which is basically one growth of knotweed, at the moment, we would need about 30 to 40 viability tests to be able to model that blob. Now, that number will decrease as we get more data in the future, we hope, but this is the situation where we are at the moment. At the on the left-hand side, the bottom picture is showing you test results where the rhizome is viable. Basically, it has failed it's worse than the unacceptable outcome. So it's probably not worth. That's probably where a test or a treatment has failed. And the top one is showing an outcome that's in between the unacceptable and the acceptable outcomes. And probably a next course of action here would be to do more testing to see if that changes your statistics a little bit. So that's basically how it works. We can look at the individual rhizome outcomes, and we can also look at statistical significance across an area. A couple of other things to note: the distribution of viability test outcomes is not normally distributed. So the process is that essentially we would be looking to have freshly extracted rhizome, which would be packaged according to a protocol that we'd provide. The samples need to be with our independent lab in Belgium if they're EU samples. The lab will then prepare the rhizomes for testing. We will get the test data and we will independently verify the outcome. And then the results would be sent to the client detailing the finding. So this is quite important. So InnoLab is not carrying the liability of giving you the determination on viable or not viable. It's simply coming up with a test outcome. The test outcome comes to us and we make the determination. Results are normally available within 48 hours and will be notified electronically. And we would confirm which of the samples are viable or non-viable and also give you a statistical prognosis. The cost within Belgium is €125 per test, which includes the plan and the protocols, the sampling, the handling of the samples by the lab, the testing regime, the data analysis, the detailed report, and statistics. Obviously, we wouldn't be including in that the packaging, any local taxes, or your dispatch costs. If PDR3 is involved in your treatment, and we find that the treatment outcome for your infestation is that it's rendered not viable, then you can take out an insurance-backed warranty, which is underwritten by a syndicate in Lloyd's. Nobody else has this warranty, and as a standard, this covers not just the costs of you retreating the site, but also the costs of any structural damage that are caused by a regrowth of knotweed after it's supposed to have been treated. So, for instance, if you treated your site, you took out the warranty, and then at some point in the future, I can't quite remember the time frame, but knotweed started poking through your tarmac and ruining your car park, then our warranty covers you fixing the car park as well as treating the knotweed. This could be quite important for some users. That's it, really. Any questions? David, there's a couple of minutes if you want to add any comments to this, particularly about dormancy and your experience of knotweed treatment over the past 20 years on I don't know how many sites.
Yeah, thanks, Paul. I mean, I just want to add just a couple of things to Paul's presentation there. It's the from leading off all the presentations that have been there this morning, our main aim going back 12 years ago when we started collaborating with Paul and the University of Reading was to find alternatives to herbicide application. And one of the crucial things that we found at the very start is trying to prove or disprove whether or not we was dead or alive after doing some trials using heat treatments. So that's where it came from originally in terms of trying to find a way of predicting whether or not he's actually dead or alive. A couple of things from the other presentations as well, which has been really good today, what was about the heat application and actually getting the core temperature to the right temperature. And that's something we spent quite a long time studying and finding out what the key figures and key components are to actually get the core temperature to the right temperature. Yeah, and in terms of rhizome depth, very, very variable. And as people have said already this morning, the depths can vary from 1 meter down to the extreme that we found is 4 to 4.5 meters when we had rhizome going down at the coal, following a coal seam. So it is very variable in terms of depth. And very hard to understand what the depth will be until it actually excavate the material out, which is another area of research which needs to be looked into, is how we can actually scan the soil or understand the actual depth of the rhizome. And again, we've actually found rhizomes at the extreme depth of 3 meters that is still viable. So the depth doesn't really tally in terms of whether it's viable or not. I mean, the whole of the rhizome we found to be viable, and we also found the root structures were also viable as well from our past experience. But yeah, that's what I need to add, Paul, I think.
Thank you very much, Paul and David. This is really the viability testing is really a crucial aspect in the treatment methods from the perspective of a site manager, but also from perspective of a developer of different techniques or methods for treatment. So it was really an interesting presentation. Actually, we are concluding this section of the treatment methods. Are there some questions for Paul and for David? Okay, so now after having seen some different methods for the treatment of the Japanese knotweed, we will end this webinar with a project that was carried out by the Public Research Center in the Walloon region in Belgium about a new value chain for the valorization of the Japanese knotweed. So, Emerald?
Yeah. Thank you. A minute. Is it okay like this?
Yeah.
Okay, thank you. Well, hello, everybody. First of all, let me thank SOILveR for inviting me to this webinar and to give me the opportunity to present a feasibility study that we carry out in 2020-2021 concerning the development of an innovative way of treatment and valorization of excavated land infested by Japanese knotweed in Wallonia. Well, before getting to the heart of the matter, let me briefly present what is ISEP. So as Marta told you, ISEP is the scientific institute working on behalf of the administration and the Walloon Minister of the Environment. Our activities are focused on environmental monitoring of all environmental compartments, such as air, surface and groundwater, industrial wastewater, soil, sediment, waste, and so on, chronic and accidental risks, and a very important topic these days, I mean the assessment of the impact of environment and pollution on human health. ISEP is also recognized as a scientific center or research organization. As such, and in parallel with the missions it carries out on behalf of the administration, ISEP is leading a lot of research projects related to these environmental thematics with diverse collaborators. Just for your information, our head office is located in Liège, in Wallonia, south part of Belgium, and we also have some colleagues working in Colfontaine, close to Mons. So, what's the origin of the Japanese knotweed project? Of course, with in mind all the problems related to Japanese knotweed management that we already discussed this morning. From ISEP point of view, we really began to face the problem of invasive plant in 2019, when a Walloon decree defined the management and traceability of procedures for excavated soil in Wallonia, and a dedicated reference guide was published outlining some guidelines for managing, namely Asian knotweed. However, no intervention or remediation obligations were imposed by the current regulation. Nevertheless, it's raised many questions about the management of these invested land, and above all, raised awareness of the need to take in charge the problem and to establish operational solution for knotweed management. And on the other end, and almost at the same time, we were approached by the SOILveR. It's a Belgium research center, expert in vegetal extraction, whose part of its research is focused on applications using plant extracts. We started, as part of its research, SOILveR sources extracts from China, but the quality and stability of are not good. Or in France, where extracts are produced from vine branches, they are of excellent quality, but very expensive for lab trials. So together, we decided to set up a feasibility study for the development of a value chain, which combines environmental management of infested land and the valorization of Japanese knotweed rhizomes by extraction of high added value substances, a value chain dedicated to Walloon region, let's say. So here are a brief description of both collaborator, ISEP, which is an expert in soil management and traceability, as I said. He has an expertise in management of polluted soil, circular economy, and is leading innovative project management. So in the Japanese Knotweed project, ISEP took in charge more specifically environmental aspects, while SOILveR is an expert in food technology extraction, the production of high added value from local plant resources, and the use of vegetal extract for diverse applications. So SOILveR took part of this thematic on the project. So actually, we were both supported by the administration, and more specifically, the interdepartment invasive species unit, which is the competent authority in Wallonia. And this unit has drawn up a decision tree which defines the action to be taken on infested land, depending on the configuration of each infested site, the potential nuisance caused by the plant, the size of the clumps or patches, and their projected evolution, future landscaping projects, and finally on whether or not it is needed to intervene, and if an intervention is required, which technique is the best to use. In this framework, 10 technical sheets are also available describing how to process regarding early eradication techniques, local eradication techniques, such as deep landfill excavation and transportation to an authorized center, crushing and tarping, and so on. And also mitigation techniques by mainly acting on the aerial part of the plant, sheep grazing, repeated grubbing, herbicides, treatments, and so on. Well, I invite you to go to see this website where the reference is written hereafter, and you can discover all these technical sheets. So here's the schematic diagram of the loop we wanted to evaluate the technical feasibility and financial viability with, as a starting point, a land to be excavated because infested by Japanese knotweed. But it must be stressed that this project was only dealing with excavated infested land. At this point, we never considered the intervention on an infested land where the remediation was not envisaged or needed, even at short term. This aspect was considered as a perspective of the project. So the loop includes a soil excavation, soil sorting in order to isolate the rhizomes from the soil and to obtain a clean soil, the vegetal extraction of high added value molecules from rhizomes, such as in resveratrol, which is a stilbene derivative, which has interesting antioxidant properties, but also other molecules of interest. The marketing of Japanese knotweed extracts to producers active in the sector of cosmetics, food supplements, or even biopesticides. And finally, the use of financial benefits to cover the cost of Japanese knotweed management. In the projected business model, the profit from this process would also be used to contribute to the fight against invasive species in Wallonia. So the relevance of the model was assessed and compared with the current situation. So the previous diagram can be divided into main stages with the extractable material as the central element. The extractable material corresponds to isolated, washed, dried, and grinded rhizomes. So we have the upstream chain, which consists of excavation of infested land, sorting of rhizomes, then preparation of the extractable material that will be transferred to the laboratory and to enter in the downstream chain from extraction of high added value molecules that are contained in the material to the marketing of Japanese knotweed extracts in the agri-food industry. At the beginning, resveratrol was the targeted molecules, but during lab trials, it appeared that other molecules of high interest were also present in Japanese knotweed extracts. So, with regard of the upstream chain, the various explorations concluded that the following sequence of steps was the most effective and operational for isolated rhizomes and in fine extractable material. After a targeted excavation of the top 25 centimeters of soil, which concentrates almost all the rhizomes, the soil is screened either on site or off site after transportation to an authorized treatment plant. And the rhizome contains in the sieving refuse are then separated from the stony fraction by hand picking, are then submitted to washing, drying, and shredding step to obtain the precious extractable material. It's important to remember that certain crucial aspects still needed to be deeply assessed, such as the quality control of soil after screening or rhizome isolation, the management of deep soil, which contains only a low density of rhizomes, and the aftercare cost, which consists on visual control for a certain period after treatment. The downstream chain includes the lab experiments aiming to obtain the Japanese knotweed extracts by hydroalcoholic maceration and extraction from the extractable material. To get a high quality extract, SOILveR, the laboratory, optimized the maceration and extraction conditions and then proceeded to an enrichment step of the extract to increase the concentration of targeted molecules. Well, it must be stressed, and it's very important that hydroalcoholic maceration and extraction conditions lead not only to the production of interested extracts, but also lead to the deactivation of rhizomes. This is also a strength of this value chain in the overall assessment of the value chain, because we all know that rhizomes elimination is very expensive, such as landfilling or thermal treatment. So the main reports we get from a field investigation in the upstream chain. So we did some investigation that were focused on two main topics. First, the estimation of the exploitable deposits in Wallonia, and two, the identification of existing techniques for separating Japanese knotweed rhizomes from infested excavated soil. The goals of this first step during field investigation were to estimate the rhizome weight in excavated infested soils, and two, to evaluate the vertical and horizontal gradients of rhizome concentrations in soil during excavation. While collecting this information is essential to obtain an estimate of the quantity of material that could be used in the in the value chain and in the extraction process. Well, in this approach, only infested soils for which excavation is unavoidable were envisaged. Of course, much larger deposits exist in Wallonia, riverbanks, roadsides, railroad tracks, natural or agricultural area. For these, the solution of excavation followed by decontamination is rarely favored for economical reasons first, but also given the risk of dissemination incurred when handling soil containing rhizomes. So our investigation took place on 2 study sites, a brownfield site that was being rehabilitated and a plot of land on which a building project is planned. The field campaigns revealed the following findings. Well, based on the lithology encountered during excavations at both sites, the rhizome network developed mainly in the first 25 centimeters of soil. Beyond this fringe, the density of rhizome decreased sharply from presence to nearly absence. In the encountered case, the horizontal dispersal of rhizomes around the clumps stopped at the edge of the clumps. That was our findings there. And regarding the vertical boundary, orange sections of rhizome were still occasionally seen at the bottom of excavation at a depth of 1.60 meters. Although excavation stopped at this depth, the primary objective of determining weight percentages as a function of depth was achieved. So the best option for rhizome separation is in excavated soil is in the top of 25 centimeters was the hand picking to isolate rhizomes. Given the maximum concentration is this horizon. All rhizome fragments, easily identifiable by the dark orange coloration, were separated for subsequent weighing. So based on the empiric data collected during our field investigation, an average load, the weight percentage, could be estimated. And according to the estimation, we found that 1 ton of excavated soil from the first top 25 centimeters contains 19.8 kilograms of rhizomes. And so the weight percentage of rhizomes in the lower soil layer is considered as negligible. So finally, the most pragmatic option for managing the remaining very lightly infested soil that has not been excavated is on-site containment. This can be achieved by laying a suitable geotextile at a depth of at least 2 meters, for example. And one of the advantages of this, apart from the fact that the soil does not need to be excavated and moved, is that it has no impact on future urban projects. So in 2020, when we started the project, the estimated costs for rhizome separation, including excavation, sieving, hand picking, and further land management, were around 12 euro a ton versus 18 euro per ton for conventional management. Costs were established on the basis of the treatment of 1,500 tons of excavated land. So additional costs are to be considered regarding the management of lightly infested soil, of course, and the land aftercare. So the main reports from field investigation in the downstream chain. So the objective of this downstream chain was to compare existing methods or techniques for extracting resveratrol from Japanese knotweed rhizomes once these are separated from the soil, washed and shredded. So the minimum indicator that SOILveR has been taken into account were extraction costs and yields, as well as the quality of the final product. At the end of extraction trials in laboratory, so the following findings and conclusions were drawn. So at the beginning of the project, resveratrol and its derivatives were the target molecules for their antioxidant properties. After laboratory trials, other high added values molecules were identified, catechin, for example, and its derivatives as an antioxidant, and amodine and other anthraquinones, which have purgative properties, for example. So the raw material, rhizomes isolated from the soil, must undergo the washing, drying, shredding to obtain the extractable material, and this operation results in a 30% loss in weight between the raw rhizomes and the extractable materials, which is then subjected to plant extraction. And of course, this value of 30% of loss depends according to the sampling conditions and the appearance of the raw rhizomes. The plant extraction method used is hydroalcoholic maceration. So the extraction parameters were optimized specifically for the rhizomes, and the well, up to now, extraction techniques that are developed by SOILveR are still protected. So if you have questions, the best way is to contact SOILveR directly. So the analysis of the knotweed extracts also showed that rhizomes sampled in spring, it was during March, they were richer than those taken in other seasons. Well, this corresponds actually to the moment when the plant wakes up, and so the rhizome contains more active molecules. Okay. So after sorry, a little bit lost. So in the case we encountered the, oh, sorry, I'm really lost. Okay, yes, sorry. So what about extraction yields? So the extraction has provided an extract representing 20% of the raw material used. This extract could be concentrated in active molecules to gain a few percent of concentration. So finally, the active molecules were concentrated on resin, and in terms of mass, after all steps, 1% of the raw material is considered to be present in the final extract. So at the end of the extraction and enrichment process, from 1 kilogram of extractable material, it's possible to obtain 10 grams of enriched knotweed extract containing 20% of active ingredients, so the targets of the molecules. And as already said, the residue obtained after the extraction step is totally inactivated for 2 reasons. It has been shown that grinding the rhizomes to 4 millimeters is sufficient to biologically inactivate rhizome fragments, and lab conditions applied during the extraction process, high pressure in a water ethanol mixture, led to the biological inactivation of Japanese knotweed rhizome. So in conclusion, the vegetal extraction process is a well-known technique that would apply to the extractable material prepared from Japanese knotweed. And the process was optimized to lead to enriched extract of high value, added value, and very good quality. So finally, we applied the findings that we made for both the value chain, upstream and downstream value chain to try it on an existing site infested by Japanese knotweed when we proceeded the field investigation. So the owner's ambition is to build an urban project after remediation. So here we have 5-acre infested land by Japanese knotweed. This is chemically contaminated. This is the red outline, and the part infested by Japanese knotweed is the yellow outline. So based on the quantity of rhizomes per square meter and the weight percentage of rhizomes encountered in the superficial soil fringe, we have estimated the presence of 20 tons of rhizomes after separation from infested land. So these 20 tons correspond to raw rhizomes, which then needed to be transformed into extractable matter with a loss of 30%. So after that, the quantity of extractable material could be estimated to 40 tons. And based on the cost of laboratory process for this quantity, excluding the cost of purchasing the raw material, the cost is estimated at between 63,000 and 77,000 for 40 tons of extractable materials. And non-optimized profit margins for marketing total extract are estimated at between at maximum 10,000 euros per ton of extractable material. And finally, sorry, I'm a little bit lost with all these figures. So, but at the end, the downstream chain profit margin is around 20,000 to 140,000 euro, which must cover the upstream chain cost. So sorry, I'm not I'm not quite comfortable with this second part of the value chain, since ISEP was involved but not deeply involved in this part of the work. So take-home message to finish my presentation. So most of the objectives set by this study have been achieved. They have led to a better perception of the proposed value chain in Wallonia, a more integrated management of the land infested by Japanese knotweed, and have opened up very interesting prospects for the extraction and marketing of rhizome extracts. For this last aspect, the results even exceeded expectation, with the possibility of joint valorization of several Japanese knotweed extracts. Well, however, some objectives were only partially achieved. For example, the quality control of the treated land after separation of the rhizomes. That being said, this led to identify the constraints and obstacles to overcome in the future, so that the sector can be more profitable and all the actors, sanitizer, treatment center, administration, and so on, can develop techniques offering all the necessary environmental guarantees. So even if the economic model still needs to be refined by optimizing certain stages in the process, the first study opens up very interesting prospects for site owners. It should be remembered that the primary objective of this entire approach is to achieve a control of this invasive plant through a structured and financially profitable value chain. Well, about financial viability, it must be remembered the existence of this French company, which is called Rhizomex, who successfully operates the value chain locally in France and since 5 years. Well, the crucial point, and that's what we did this morning, is make the link between all the stakeholder. And well, I think that's all the presentation of this morning will bring inputs in the value chain I presented here.
Okay.
Just an information, if I can, just to save the date, because in November, ISEP is organizing with the invasive species unit of the administration a thematic day dedicated to Japanese knotweed. So you can save the date. It will be in Namur. And if you are interested, you can contact my colleague, Amandine Staren, to have more information and to receive more information if you are interested in this very interesting day.
Okay. Thank you very much, Emerance. I'm afraid that we are running behind the schedule, so we'll skip the final discussion, but I think that during the presentations and the question session, some of you had the opportunity to ask your questions and some point of view. So I'd like to say a big thank you to all the speakers for your valuable contribution today. It was really very interesting to have this overview from different countries on different treatment methods. So for the audience, thank you also for your patience. I hope that you found this webinar and the topic really interesting, and it was also useful for you to learn from different experiences and methods that were tested and developed in different countries. So touch with SOILveR, because in September, October, and November we will organize, and we plan to organize other coffee breaks or webinars on different topics. So you'll receive the invitation to attend those. So thank you again very much, and have a nice day ahead. Thank you. Bye. Okay, so